in the adult, the ear forms one anatomic unit serving both hearing and equilibrium. In the embryo, however, it develops from three distinctly different parts: 1. The external ear, the sound collecting organ. 2. The middle ear, a sound conductor from the external to the internal ear. 3. The internal ear, which converts sound waves into nerve impulses and registers changes in equilibrium. Internal ear, the first indication of the developing ear, can be found in embryos of approximately 22 days as a thickening of the surface ectoderm on each side of the rhombencephalon, Fig.1. These thickenings, the otic placodes, invaginate rapidly and form the otic or auditory vesicles, otocysts, Fig.2. Cells from the otocyst differentiate and form ganglion cells for the statoacoustic, vestibulocochlear, ganglia. During later development, each vesicle divides into 1. A ventral component that gives rise to the saccule and cochlear duct. 2. A dorsal component that forms the utricle, semicircular canals, and endolymphatic duct, Figs 3 to 6. Together, these epithelial structures form the membranous labyrinth. Saccule, cochlea, an organ of Corti. In the 6th week of development, the saccule forms a tubular outpocketing at its lower pole. This outgrowth, the cochlear duct, penetrates the surrounding mesenchyme in a spiral fashion until the end of the 8th week, when it has completed 2.5 turns, Fig 3d. In the 7th week, cells of the cochlear duct differentiate into the spiral organ of Corti that transduces a sound vibrations into electrical signals for hearing. A connection between the cochlear duct and the remaining portion of the saccule is maintained but confined to a narrow pathway, the ductus reuniens. Mesenchyme surrounding the cochlear duct soon differentiates into cartilage, Fig 4a. In the 10th week, this cartilaginous shell undergoes vacuolization, and two perilymphatic spaces, the scala vestibuli and scala tympani, are formed, Fig 4bc. The cochlear duct is then separated from the scala vestibuli by the vestibular membrana and from the scala tympani by the basilar membrane. The lateral wall of the cochlear duct remains attached to the surrounding cartilage by the spiral ligament, whereas its median angle is connected to and parallelly supported by a long cartilaginous process, the modiolus, the fiature axis of the bony cochlea. Initially, epithelial cells of the cochlear duct are alike. With further development, however, they form two ridges, the inner ridge, the future spiral limbus, and the outer ridge. The outer ridge forms one row of inner and 3 or 4 rows of outer hair cells, the sensory cells of the auditory system. They are covered by the tectorial membrane, a fibrillar gelatinous substance attached to the spiral limbus that rests with its tip on the hair cells, Fig 5. The sensory cells and tectorial membrane together constitute the organ of Corti. Impulses received by this organ are transmitted to the spiral ganglion and then to the nervous system by the auditory fibers of cranial nerve V3. Utricle and semicircular canals. During the 6th week of development, semicircular canals appear as flattened outpocketings of the utricular part of the otic vesicle, Fig 6ab. Central portions of the walls of these outpocketings eventually oppose each other and disappear, giving rise to 3 semicircular canals, Fig.8. Whereas one end of each canal dilates to form the crus ampullar, the other, the crus non-ampullar, does not widen. Because 2 of the latter type fuse, however, only 5 crura enter the utricle, 3 with an ampulla and 2 without. Cells in the ampullae form a crest, the crista ampullaris, containing sensory cells for maintenance of equilibrium. Similar sensory areas, the maculae acousticae, develop in the walls of the utricle and saccule. Impulses generated in sensory cells of the cristae and maculae as a result of a change in position of the body are carried to the brain by vestibular embryos of cranial nerve 8. During formation of the otic vesicle, a small group of cells breaks away from its wall and forms the statoacoustic ganglion, Fig.2c. Other cells of this ganglion are derived from the neural crest. The ganglion subsequently splits into cochlear and vestibular portions, which supply sensory cells of the organ of Corti and those of the saccule, utricle, and semicircular canals, respectively. Middle ear, tympanic cavity and auditory tube. The tympanic cavity, which originates in the endoderm, is derived from the first pharyngeal pouch, Fig. 7. This pouch expands in a lateral direction and comes in contact with the floor of the first pharyngeal cleft. The distal part of the pouch, the tubotympanic recess, widens and gives rise to the primitive tympanic cavity, and the proximal part remains narrow and forms the auditory tube, eustachian tube, Figs 8, through which the tympanic cavity communicates with the nasopharynx. Ossicles. The malleus and incus are derived from cartilage of the first pharyngeal arch, and the stapes is derived from that of the second arch, Fig. 9a. Although the ossicles appear during the first half of fetal life, they remain embedded in mesenchyme until the 8th month, when the surrounding tissue dissolves. The endodermal epithelial lining of the primitive tympanic cavity then extends along the wall of the newly developing space. The tympanic cavity is now at least twice as large as before. When the ossicles are entirely free of surrounding mesenchyme, the endodermal epithelium connects them in a mesentery-like fashion to the wall of the cavity, Fig. 9b. The supporting ligaments of the ossicles develop later within these mesenteries. Because the malleus is derived from the first pharyngeal arch, its muscle, the tensor tympani, is innervated by the mandibular branch of the trigeminal nerve. The stapedius muscle, which is attached to the stapes, is innervated by the facial nerve, the nerve to the second pharyngeal arch. During late fetal life, the tympanic cavity expands dorsally by vacuolization of surrounding tissue to form the tympanic antrum. After birth, the epithelium of the tympanic cavity invades the bone of the developing mastoid process, and epithelium-lined air sacs are formed, pneumatization. Later, most of the mastoid air sacs come in contact with the antrum and tympanic cavity. Expansion of inflammations of the middle ear into the antrum and mastoid air cells is a common complication of middle ear infections. External ear, external auditory meatus. The external auditory meatus develops from the dorsal portion of the first pharyngeal cleft. At the beginning of the 3rd month, epithelial cells at the bottom of the meatus proliferate, forming a solid epithelial plate, the meatal plug, Fig. 7. In the 7th month, this plug dissolves, and the epithelial lining of the floor of the meatus participates in formation of the definitive eardrum. Occasionally, the meatal plug persists until birth, resulting in congenital deafness. Eardrum or tympanic membrane. The eardrum is made up of 1. An ectodermal epithelial lining at the bottom of the auditory meatus. 2. An endodermal epithelial lining of the tympanic cavity. 3. An intermediate layer of connective tissue, Fig. 9b, that forms the fibrous stratum. The major part of the eardrum is firmly attached to the handle of the malleus, and the remaining portion forms the separation between the external auditory meatus and the tympanic cavity. Auricle. The auricle develops from 6 mesenchymal proliferations at the dorsal ends of the first and second pharyngeal arches, surrounding the first pharyngeal cleft, Fig. 10. These swellings, auricular hillocks, 3 on each side of the external meatus, later fuse and form the definitive auricle. Because formation and fusion of the auricular hillocks is complicated, developmental abnormalities of the auricle are common. Furthermore, because the hillocks are derived from neural crest cells, external ear defects are often associated with malformations in other organs derived from neural crest cells, such as the face, skull, and heart. Initially, the external ears are in the lower neck region, but growth of the mandible posteriorly and cranially moves the external ears to the side of the head at the level of the eyes. Hearing is dependent on sound waves that cause vibrations in the tympanic membrane, which is held tightly like the surface of a drum by the tensor tympani muscle. If too loud a sound, causing potentially damaging vibrations, occurs, the tensor tympani together with the stapedius muscle stretches the membrane even tighter to prevent it from vibrating too forcefully. Once the tympanic membrane vibrates, it causes movement of the ear ossicles, the malleus, incus, and stapes that serve to amplify the force of the sound wave and to transmit the pressure of the wave to the cochlea through the oval window, Fig. 8. Amplification results from 2 factors, 1. the large size difference between the tympanic membrane, 55 mm, and the oval window, 3.2 mm, where the stapes attaches, results in greater energy produced by the membrane to be transferred and amplified at the oval window. 2. Because of its shape, the malleus acts like a lever to increase the force received by the stapes, Fig. 8. Pressure produced by movement of the stapes at the oval window irradiates a fluid wave in the cochlea that is balanced by movement of the round window. The fluid wave moves small regions of the basilar membrane, and the location of these regions is determined in part by the amplitude, loudness, and frequency, pitch, of the wave. Near the oval window, the basilar membrane is attached by shorter stiffer fibers to the side of the cochlea. Farther along the cochlea, the fibers are longer and more flexible. Based on these fiber characteristics and the frequency of the wave reaches a point where it resonates with the fibers and moves the basilar membrane. This movement is sensed by adjacent hair cells that send impulses back through nerve fibers of the acoustic segment of the statoacoustic nerve. High frequencies, high pitch, are heard near the oval window where fibers connecting the basilar membrane are shorter and stiffer. Lower frequencies are heard farther up the cochlea where the fibers are longer and more flexible. Hearing loss and external ear abnormalities. Congenital hearing loss may be due to abnormalities of the hair cells or auditory nerve ganglia, sensorineural loss, or to structural defects in the external ear canal, eardrum, or ear ossicles, conductive hearing loss. 50% of cases are genetic and may involve autosomal dominant or recessive or X-linked genes. In Treacher-Collins syndrome, malformations in the external ear and ear canal and the ear ossicles can all be involved in causing varying degrees of conductive hearing loss. In Down syndrome, hearing loss may be caused by sensorineural and/or conductive abnormalities. Most cases caused by other factors are characterized by sensorineural defects. These factors include prenatal infections, rubella, cytomegalovirus, herpes simplex, prematurity, and maternal diabetes. Accutane, retinoids, can cause both types of deafness. The most common ear ossicle to be involved in conductive hearing loss is the stapes, and this fact may be due to its unique origin because its footplate is derived from neural crest cells and paraxial mesoderm. Neural crest cells direct formation of the footplate and of the oval window. Without directive signals from the neural crest, neither the footplate nor the oval window develops properly. Stapes fixation, ankylosis, is the most common abnormality and usually involves both ears. External ear defects are common. They include minor and severe abnormalities, Fig.11. They are significant from the standpoint of the psychological and emotional trauma they may cause and for the fact that they are often associated with other malformations. This association is due in part to the fact that external ears are derived from neural crest cells, and this cell type contributes to development of many other structures, including the face, skull, and heart. Thus, external ear defects serve as clues to examine infants carefully for other abnormalities. All of the frequently occurring chromosomal syndromes and most of the less common ones have ear anomalies as one of their characteristics. Preauricular appendages and pits, Fig.11 CD, are skin tags and shallow depressions, respectively, anterior to the ear. Pits may indicate abnormal development of the auricular hillox, whereas appendages may be caused by accessory hillox. Like other external ear defects, both are sometimes associated with other malformations. Summary: the ear consists of three parts that have different origins but that function as one unit. The internal ear originates from the otic vesicle, which in the fourth week of development detaches from surface ectoderm. This vesicle divides into a ventral component, which gives rise to the saccule and cochlear duct, and a dorsal component, which gives rise to the utricle, semicircular canals, and endolymphatic duct, Figs 3, 6, and 8. The epithelial structures thus formed are known collectively as the membranous labyrinth. Except for the cochlear duct, which forms the organ of Corti, all structures derived from the membranous labyrinth are involved with equilibrium. The middle ear, consisting of the tympanic cavity and auditory tube, is lined with epithelium of endodermal origin and is derived from the first pharyngeal pouch. The auditory tube extends between the tympanic cavity and nasopharynx. The ossicles, which transfer sound from the tympanic membrane to the oval window, are derived from the first, malleus and incus, and second, stapes, pharyngeal arches, Fig 9. The extemal auditory meatus develops from the first pharyngeal cleft and is separated from the tympanic cavity by the tympanic membrane, eardrum. The eardrum consists of 1. An ectodermal epithelial lining. 2. An intermediate layer of mesenchyme. 3. An endodermal lining from the first pharyngeal pouch. The auricle develops from 6 mesenchymal hillocks, Fig. 10, along the first and second pharyngeal arches. Defects in the auricle are often associated with other congenital malformations.